afternoon, everyone. So today's lecture will be on synaptic neurotransmission. It's very simple. It's a simple topic. I think it's simpler than the previous lecture, maybe. So let's talk about synaptic neurotransmission. So basically, we'll start with defining the synapse, what a synapse is. Then we'll talk about the difference between a chemical and an electrical synapse. Then the next part, we will talk about the sequence of events that happens during chemical synaptic neurotransmission. Then finally, we'll talk about summation of the synaptic potentials. Okay. So everyone, you can hear me? Or do I have to raise the...
Can you raise your voice?
Okay. So let's start. To define what a synapse is. And so a synapse is basically the functional contact between a sending neuron and a receiving neuron. So as you can see here, we have this neuron here, which is a sending neuron, okay? So you can see the cell body, the axon, and here we have the axon terminals, okay? And here we have the receiving neuron, okay? And so here we have the dendrites of the receiving neuron. And so the sending neuron will be communicating with the postsynaptic neuron or the receiving neuron at this site here, which is what we call a synapse, okay? And so the sending neuron in this case would be called presynaptic neuron, and the receiving neuron would be called postsynaptic neuron, okay? So it's very simple. So a synapse basically is the functional contacts, and the main purpose is to transfer the information from the presynaptic neuron into the postsynaptic neuron, okay? Now, there are different types of synapses. The main ones are either chemical synapse or an electrical synapse, okay? The main difference is that a chemical synapse is the most popular, so the most common throughout the nervous system is the chemical synapse, okay? Okay, so here what you see, the green here, the green synapse is the chemical synapse, and the blue is the electrical synapse. The chemical synapse is the most common, okay? It is characterized by the asymmetrical morphology. So please next time be on time, huh? Because we don't want to distract the... Okay. So a chemical synapse is the most common, and it's mainly characterized by the asymmetrical morphology. What do we mean by asymmetrical morphology? Is basically the machineries on the presynapse is different to the machineries on the postsynapse. As you can see here, that on the presynapse we have synaptic vesicles, which contains the neurotransmitters, which we will talk about in the upcoming slides. And on the postsynapse, we have the receptors, okay? So different machineries exist on the presynapse to the postsynapse. So as asymmetry. The flow of information tends to be unidirectional. So it's always from the presynapse into the postsynapse, okay? Also, there is a gap between the presynapse and the postsynapse, which we call synaptic cleft. So they are not physically in contact, okay? The information is mediated through the release of the chemical agents, the neurotransmitters. And so this is why it's called a chemical synapse. And so because of the release of the neurotransmitters, and because there is a gap between the presynapse and the postsynapse, there is usually a slight delay in transferring the information, okay? Very slight delay. Now, with the electrical synapse, it's not rare. There are electrical synapses, but less common than the chemical synapses, okay? It is characterized by symmetrical morphology. As you can see here, the presynapse is the same as the postsynapse in terms of the machineries. The flow of information can be bidirectional, okay? Unlike the chemical synapse, it's unidirectional. It's very fast. The transfer of information is very fast, and it's mediated mainly by the flow of ions, okay? And also, electrical synapses are physically contacted, okay, through these channels here, which are called gap junctions, okay, which are made up of proteins like connexins, etc. So these electrical synapses are mainly found in circuits which require synchronization. Anybody is familiar with the word synchronization? Exactly. So synchronization is when signals are required to be, at the same time. So it will help in the synchronization of the signal. For example, in circuits which play a role in, mediating our breathing patterns, we have electrical synapses, okay? Because they are required to help the neurons to fire in synchronicity, okay? So this is the main difference between the chemical and the electrical synapse. So the rest of the lecture, we will focus on the chemical synapse, okay? So we won't really talk about the electrical synapse. Now let's talk about the major steps of the chemical synaptic neurotransmission. So the first step is that, as we learned from the previous lecture, okay, so this is the presynaptic neuron, and here we have the postsynaptic neuron, okay, or a muscle cell or whatever. Now, depolarization of the presynaptic neuron, so here we have an action potential coming down to the synaptic terminal. So depolarization of the presynaptic terminal would basically activate or open a calcium voltage gated channel, okay? So opening of the voltage gated calcium channels would allow the movement of calcium to go from outside to inside, okay? Do you know why? From outside to inside? Because of the concentration difference. So we know that calcium is, the concentration of calcium is higher outside than inside. So calcium will go in. Now, calcium inside the, synaptic terminal will trigger the release of the synaptic vesicles, okay? So we have these synaptic vesicles, which contains the neurotransmitters. So the entry of calcium will trigger the fusion of the synaptic vesicles with the presynaptic terminal, okay? Now we have the neurotransmitters here are being released in what is this called? Synaptic cleft. Very good. So we have the neurotransmitters in the synaptic cleft. What these synaptic neurotransmitters will do is that they will bind into into their respective receptors on the postsynaptic membrane. Okay, is that clear? So once they are bound, they will open the postsynaptic receptors, allowing flow of ions. And as we will see in the upcoming slides, depending on which ion is entering, it could be either excitatory or inhibitory, okay? So it's not always excitatory. It depends on many things, including the neurotransmitter, the type of channel, the type of ions going inside. So now, after binding, now what will happen to the neurotransmitters? They will be recycled again. So where they will be uptaking into the presynaptic terminal, okay, and being recycled, okay, where they will be again refilling synaptic vesicles for the next round of the synaptic neurotransmission. Okay, is that clear? Good. Let's go to the Now let's talk about each individual features in a bit more details, okay? So let's talk about, so firstly, we will, just, talk about the neurotransmitters. So neurotransmitters, you will learn more about these, the different types, I think in tomorrow's lectures with Dr. Hamdan, okay? But here, what you need to know is that the neurotransmitters are the chemical agents which the chemical synapse uses to transfer the information. Now, they can be categorized into two main categories, okay? So we have the small molecule neurotransmitters and we have the neuropeptides. The small molecule neurotransmitters, as the name suggests, they are small, they are individual amino acids, okay? Like glutamate, GABA, it's considered the small molecule neurotransmitter. And we have the neuropeptides, they are large molecules, they are made up of 3 to up to 36 amino acids, they are very large. Now, the small molecule neurotransmitters, as you can see here, so they are made up, we said, of what? The small molecule neurotransmitters. Individual amino acids, okay? So they and locally within the presynaptic terminal, okay? However, the enzyme that contribute into making the precursors for these small molecule neurotransmitters are available in the presynaptic terminal. But the enzyme that contribute into the synthesis of these small molecule neurotransmitters are transported from the soma, from the cell body. So here we have the enzymes, okay, which are coming to here to help in the synthesis of these small molecule neurotransmitters, okay? Whereas with the neuropeptides, they are made up in the soma, in the cell body, packaged, and then get transported to the presynaptic terminal, okay? So that's one difference. Another difference is that the small molecule neurotransmitters, they usually mediate fast action, whereas the large ones, usually they mediate slow, longer lasting action, okay? So this is very general overview of the neurotransmitters. Now, what will happen is that when the presynapse is depolarized, so we have these synaptic vesicles, they have neurotransmitters, they are being stored in what we call reserve pool, okay? Now, when the presynaptic terminal is depolarized, what will happen is that we know what will happen. So anyone? So after depolarizing the presynaptic terminal, opening of the voltage gated calcium channels, entry of calcium will trigger what? Fusion of the synaptic vesicle. Very good. So a synaptic vesicle will go through the process of docking, priming, and then fusion to release the neurotransmitters. And finally, so what you can see here is that after fusion, the added membrane, because this is an extra membrane from the synaptic vesicle, will be endocytosed for recycling, okay? And then will be filled again with the neurotransmitters, okay? Now, calcium is very important in this process, okay? So without calcium, there would be no release of neurotransmitters. And scientists did confirm this by multiple experiments, okay? So in the lab, we have dyes which you can use to stain calcium, okay? Which bind calcium, and it can show you the profile of calcium. What scientists did, as you can see here, that when injecting calcium from outside, okay, exogenously injecting calcium into the neuron, you can see that in the presynaptic neuron that there will be, they found that this calcium induces a postsynaptic membrane potential in the postsynaptic neuron, okay? So this confirms that calcium is very important for neurotransmitter release, okay? Another experiments they did is that basically here we have the presynaptic membrane potential, and here we have the postsynaptic membrane potential, the receiving neuron. By what they did is that basically when they injected the presynaptic terminal, presynaptic neuron with the calcium chelator. You know what a calcium chelator is? What?
Binds calcium.
Very good. It binds calcium and it will buffer calcium. It basically, it will buffer calcium, okay? So it binds calcium and so calcium won't be able to perform its function, okay? So when they injected the calcium chelator in the presynaptic terminal, what they found is that this basically canceled this basically cancels the postsynaptic potential, okay? So this again confirms that calcium is very important in order to release neurotransmitters and induce postsynaptic potential. Is that clear? And I would like to show you, so here I have, okay. Okay. So I would like to show you this, which I did by myself. So I do experiment with calcium, okay? And so we have neurons, which we differentiate from stem cells, okay? And we basically load these neurons with calcium dyes. And when we stimulate this, I'll show you, you can see that the release of calcium, which is very nice, okay? So I did this here actually in Saudi. It's not like, okay. So you can see here, so we have neurons, okay, which we differentiate from stem cells. Okay, so you can see that these neurons we differentiate from stem cells, then we load with the calcium dyes. And so what I did is I basically added a stimulus at time frame 20 to see if that can depolarize the neurons, okay? And what you see is this. Ooh, look at that. So this is the profile of calcium. Okay, so when we added the stimulus, you can see that this resulted in the depolarization of neurons. As we can see that this induced a huge release of calcium, okay? And so calcium is really very important when it comes to synaptic neurotransmission. Okay, so can we go back to the, sorry. Good. Any questions so far? Okay, good. Okay, so what you're looking at here, sorry, how do you come up with annotations? Okay, good. Thank you so much. Okay, so what you're looking at here, believe it or not, this is a synaptic vesicle, okay? And look at all of these proteins expressed on these synaptic vesicles. Each of these proteins, they do have important functions when it comes to all the steps of the synaptic the docking, the priming, and the fusion of the synaptic vesicle. One protein of interest would be, for example, synapsins, as an example. Synapsin is very important to connect the synaptic vesicles with each other, and thus will be tethered as a network in the reserve pool, okay? Phosphorylation of synapsins by kinases would help in the dissociation of the synaptic vesicle, okay? And so synapsin is very important. Also, some of the proteins which are very important for priming and fusion of the synaptic vesicles are the snares proteins. So, for example, synaptobrevin, okay, which is expressed on the synaptic vesicle, okay, and also we have synaptotagmin, which is expressed on the synaptic vesicle. On the membrane of the presynaptic terminal, we have syntaxin and SNAP25. So priming is basically the formation of a complex between these snares proteins. As you can see here, they are forming a complex, okay? And what scientists found is that when calcium enter into the presynaptic terminal, it will be binding to the synaptotagmin, okay? So calcium binds to which protein? Synaptotagmin, and so synaptotagmin would trigger the fusion of the synaptic terminal, of the synaptic vesicle with the membrane of the presynaptic neuron to release the neurotransmitters. This is just to show you that it is, more deep than you think. It's a complicated process, okay? Any questions so far?
Doctor?
Yeah.
Synaptotagmin, you said it's for the fusion when the calcium binds to it. You said something about the priming, I think.
Mm-hmm.
Which one of these proteins would help with the priming?
The priming is basically by making the complexes. Calcium would be important for also priming and would be important for example, activating the kinases, which will be phosphorylating synapsins, okay, to dissociate the synaptic vesicles. And so the forming of the complexes by interacting, by syntaxins on the presynaptic membrane with the synaptobrevin and SNAP25 with synaptobrevin would be basically contributing into the priming. Now, calcium binding to synaptotagmin would bring the synaptic vesicle very close with the membrane, and so thus this would lead to the fusion and the release of the neurotransmitters.
So the priming is all of these proteins, but the fusion is the calcium when
yes, but priming is more complicated as well because it involves other proteins, okay? As you can see here, so multiple proteins are really involved, but I don't want to go into too much details and talk about each protein. If you are interested, you can learn on your own, okay? So you can see the number of proteins that are involved in priming, in docking, in mobilization, etc., and even endocytosis, which I haven't mentioned, okay? But I just wanted to make it, just simpler, okay? Any questions? Yeah? No. Just focus on the ones that I mentioned, okay? Okay, so upon the release of the neurotransmitters, what will happen? We will have the neurotransmitters binding to the receptors on the postsynaptic membrane. And so these neurotransmitter receptors, they have binding sites for neurotransmitters, and they are categorized again. I think Dr. Hamdan will talk more about this, but here showing you the major categories. So they are either ionotropic receptors or metabotropic receptors. Ionotropic receptors are basically open when bound by neurotransmitters to allow the passage of ions. It's very straightforward. So neurotransmitters binding to them would open them up to allow the passage of ions. So they are linked to an ion channel, okay? And so examples is that acetylcholine binding to a cholinergic receptor would be inducing, you know, a postsynaptic potential, or glutamate binding to MDA or the AMPA receptors would be generating an excitatory postsynaptic potential. Whereas GABA, which is again can bind to ionotropic GABA receptors, can induce what? An inhibitory postsynaptic potential. Okay, is that clear? Now with the metabotropic receptor, it's different. It's okay, so glutamate, for example, binding to the metabotropic glutamate receptor would activate a G protein. Okay, so it's G protein coupled receptor. And so this G protein can either directly interact with an ion channel, as you can see here. So here we have a metabotropic receptor. So activation by binding of the neurotransmitter would activate a G protein, okay, G protein dissociating into the different subunits, and then either directly interact with an ion channel, as you can see here, or indirectly via, you know, second messengers like calcium or IB3, as I'm very sure you are familiar with these pathways, right? Anyway, I think Dr. Hamdan will cover this again in the next, but this is just to show the major categories. And also again, GABA binding into a GABAergic metabotropic receptor would induce what? What? Inhibitory, or is it excitatory? Inhibitory. So GABA binding into a GABAergic metabotropic receptor would induce an inhibitory postsynaptic response. The difference between metabotropic and ionotropic receptors is that the metabotropic receptors usually mediate much slower action and longer lasting action, okay? Whereas with the ionotropic receptor, binding of neurotransmitters to the ionotropic receptors mediate a fast postsynaptic response, okay? Now, after binding to the receptor and activating the channel, what will happen? Okay, so this is this stage. Now, in order to explain this, let's take the neuromuscular junction as an example. So the neuromuscular junction is basically the synapse between a motor neuron. So you can see here a motor neuron synapsing with a muscle cell. Okay, so this is what we call a neuromuscular junction. And scientists have been studying the neuromuscular junction because it's highly accessible. It's easy to study, okay? So we'll take the neuromuscular junction as an example to, you know, talk about what happened after binding of the neurotransmitter to the postsynaptic muscle or postsynaptic neuron in general. Okay, so as you can see here, so we have, this is the neuromuscular junction. So here we have the postsynaptic cell, which is it a neuron or a muscle? It's a muscle in this case, because this is the neuromuscular junction. And here we have the presynaptic terminal. Now, this is a cholinergic neuron, so it does have the neurotransmitters in the presynapse is acetylcholine, okay? So inside the presynaptic terminal, we have acetylcholine. On the muscle cell, okay, on the membrane of the muscle cell here, the receptors are cholinergic receptors, okay? Are they ionotropic or metabotropic? Ionotropic, they are ionotropic, okay? Now, also what we have on the postsynaptic membrane enzymes, which are called acetylcholinesterase, okay? So they are called acetylcholinesterase, which are known to break down acetylcholine. Now, the receptor, the acetylcholine receptor is permeable to both sodium and potassium, okay? So any volunteer here to explain what will happen after depolarization of the presynaptic terminal?
It would open up the voltage-basic calcium channel.
Very good.
And then it would bind to the proteins in the vesicles.
Okay, very good.
Making it fuse to the, what do you call it, the presynaptic membrane.
Very good.
And it would go to the synaptic cliff.
Okay.
And these neurotransmitters, they would bind to, what do you call it, the receptors in the muscle cell.
Okay.
And after, and then it would, and the ion flow would start. And
Okay.
after the acetylcholine finishes, the acetylcholinesterase would break it down and go recycle it.
Okay, very good. Excellent. What's your name?
Tala.
Very good. Can you clap for Tala? Very good. But we're missing a detail here. Now, acetylcholine will be binding to the cholinergic receptor, right? So here, you can see it binding. Now, we said that the cholinergic receptor is permeable to what? To sodium and potassium. So which one is going to go in? Is it sodium or is it potassium? How do you know? Good. But there is a little bit of more detail here. Okay. Now, what you learned in the previous lecture is mainly examples on channels which are selective to a particular ion. Okay. And so as you can see here, so in yellow is the potassium, the channels which are selective for potassium. In red, it's the ones that are selective for sodium. Okay. Now, bear with me. There is something called the reversal potential, which basically means it's the voltage at which the current would be reversing from outward to inward. Okay. So for example, if the channel, okay, is mainly selective, for example, only selective to potassium, as in this case, the reversal potential would be the same as the equilibrium potential for potassium. You remember the equilibrium potential, isn't it? You do. So the equilibrium potential for potassium, we know that it's around negative 100 millivolt, right? And so if the membrane, let's say, is at the resting state of negative 50, what do you think will happen to potassium? Is it going to be in or out? Is it inward or outward when the membrane is at negative 50 resting membrane? Outward, isn't it? Why?
Because the equilibrium is higher than negative.
Very good. So it must be going out in order to reach its equilibrium potential. And a nicer way to really confirm this is by calculating the ionic driving force. So which is the negative 100. Sorry, so the membrane potential would be negative 50. Minus 100 would be positive. So what is the ionic driving force? It's positive 50, isn't it? So the sign is positive, meaning it's an outward. And this is a cation, okay? If it's an anion, positive means it's inward, okay? Now, if the membrane was at negative 150, what do you think is gonna, what do you think the movement of potassium would be? It will be N, okay? And so in this case, as you can see here, that the reversal potential for the potassium selective channels is equal to the equilibrium potential for potassium. Is that clear? Clear? Now, sodium is the same thing, okay? So basically what we said is that when so now let's apply this to the cholinergic receptor, which we said permeable to both sodium and potassium. In this case, what scientists found is that the reversal potential for the cholinergic receptors is 0 millivolts. Okay, so 0 millivolt, as you can see here, is the reversal potential. Okay, and so we know that the equilibrium potential for potassium is negative 100 millivolts, isn't it? And we know that the equilibrium potential for sodium is positive 70, right? Now, if we take this equation here, which is the end plate current, so end plate, so when we talk about the muscle, we call it an end plate, okay? So end plate current, EPC, which is the current that's going to the postsynaptic muscle cell, okay? Equals, which is like we explained in the previous lecture, conductance. Conductance in this case is the conductance is based on the synaptic neurotransmitter release, which is acetylcholine, binding to the cholinergic receptor. Is that clear? So depending on how much the amount of the acetylcholine present on the, so depending on how much, so these are not voltage gated, right? Are they voltage gated, these ones? They are not voltage gated. They are ligand gated channels, isn't it? So depending on the amount of the acetylcholine and the receptors that are activated, they would be opening, isn't it? And so this represents the conductance, okay? So it's different. We are not talking about voltage gated channels here. Is that clear? We're talking about ligand gated channels. So conductance in this case is based on the neurotransmitter release of acetylcholine and the receptors that are bound to it and opened, okay? Now, this is the ionic driving force. So it's the membrane potential minus the E reversal. Now, let's take this case in here. Now, we said that the reversal potential for the end plate is what? 0 millivolt, okay? 0 millivolt is what? Come on, guys. 0 millivolt is what? Huh? Very good. It's the reversal. What do we mean by the reversal potential or equilibrium? What do we mean by that?
That's the point where both of them are in equilibrium with each other.
Very good. So it is basically the voltage at which it will be defined whether the current would be inward or outward. Because remember, this is the cholinergic receptor, which is permeable to both. It's not only to sodium or potassium, to both, okay? And so when the membrane is at rest, which is negative 90 millivolt, okay? So what do you think is gonna be, is it sodium going in or potassium going out? Sodium going in, why? Because negative, okay, so, okay, very good. But if we, for example, apply it to the equation that we have, what do you think is going to happen? So the membrane potential is what? In this case, the resting membrane potential. Minus 90, okay? What is the reversal potential? 0. So what is the ionic driving force? Okay. Negative 90. When we see negative, what does it mean? Huh? Inward. So this means which ion is going in? Sodium is going to go in. Okay. And then at 0 millivolt would be, you know, equaling to each other. Okay. But now if the membrane was, let's say, at 1 millivolt, okay, which ion will be moving? It's more of a potassium, okay? Because if you put in here 1 minus 0 will be positive 1. So it will be an outward potassium current, okay? Is that clear? Okay, and so this is this is the conclusion here. This applies to the end plate potential or current to the cholinergic receptor on the postsynaptic membrane, which is permeable to both sodium and potassium. So please don't mix it up with the voltage gated channels, which I explained in the previous lecture. Okay, so now, as we learned that at negative 100 millivolt, there will be no movement of potassium because it's, again, this is the equilibrium potential for potassium. And so when the acetylcholine is bound to the cholinergic receptor, this will allow the movement of which ion? Which ion? Sodium, okay? And so sodium will be going in. So sodium, so this is current, in the plate current. And current is usually represented when it's down, it means that the ion is moving to the inside. It's an inward current. And this is an end plate potential, okay? So it's different, it's how they represent them on graphs, okay? So current, usually, I know it's confusing, but current usually, when it's down, it means it's going inward. When it's up, it means the movement is outward, okay? It's different to the potential. So when the sodium is going inside the neuron, this creates an end plate potential until it reaches a 0 millivolt, which we said 0 millivolt what? The reversal potential, very good. So this is the reversal potential, and so at positive 70 millivolt, which ion would be moving? It's potassium is gonna move out, okay? Is this clear? Because I see dull faces in here. Is this clear? Okay, very good. So as you can see here, so this graph summarizes it. So this is the zero reversal. So this is the reversal potential. And so this part here, when it's negative, okay, when the membrane is negative, it's basically depolarizing, okay? Is this clear? When it's positive, it's hyperpolarizing, which basically by the outward movement of the potassium. Yes.
So at positive 70, potassium moves out, so we can reach 0 volt.
No, to reach the equilibrium potential for potassium. And remember, we are talking here about the cholinergic receptor, and conductance is based on the neurotransmitters. It's not voltage gated channels. So the availability of the neurotransmitters and the when the channel are activated, okay, so this is what we are talking about. So the neurotransmitter has to be bound to the channel in order for the either sodium or potassium to move, okay? So depending on the membrane potential of the postsynaptic muscle cell. Yeah.
This channel in the muscle cell, that it's selected for potassium and sodium.
Yes, so here we are talking about the end plate. So this is what I said. We are gonna talk about the neuromuscular junction as an example. Okay, and in the neuromuscular junction, the end plate, this is what they call it, the postsynaptic muscle cell, we have the nicotinic cholinergic receptors, which are permeable to both sodium and potassium upon binding of the acetylcholine. Is this clear? Good. Now, so assuming that we have the muscle cell at rest, the resting membrane potential for a muscle cell is around what? It's similar to neurons. Around negative 80 millivolt or so, okay? And so when the neurotransmitter acetylcholine is being released from the presynaptic terminal, and acetylcholine is bound to the cholinergic receptor, which ion will be moving? Sodium. Sodium will be moving, isn't it? And so entry of sodium, okay, and creating an end plate current, okay, which if it's good enough, it will be stimulating or opening up the voltage gated sodium channels, okay? And this will lead into the depolarization of the muscle, making the muscle contract. Okay, is this clear? Good. Now, after the neurotransmitters, done their job, what will happen is that the neurotransmitter will be recycled, where they will be broken down by the enzyme acetylcholinesterase into choline and acetate, and will be reuptaken again to the presynaptic terminal, okay, where they will be synthesized again to acetylcholine and filling the synaptic vesicles again, ready for another round of synaptic neurotransmission. Okay, so I won't go into much details with the cycle of the neurotransmitters, because, as I said, you will learn more in the next lectures. Now, an example of a disease where the synaptic neurotransmission is disrupted is the myasthenic syndromes. Okay, in those patients, there is generally skeletal muscle weakness, and there are, and it's an autoimmune disease. Okay, and can be classified into two syndromes. There is the Lambert-Eaton myasthenic syndrome. Okay. And there is the myasthenia gravis. Okay, in both conditions, there is a disrupted synaptic neurotransmission at the neuromuscular junction. The difference is that in the Lambert-Eaton myasthenia syndrome, the antibodies target the calcium voltage-gated channels. Okay, so when the calcium voltage-gated channels are being distracted or destroyed, what do you think will happen? No synaptic release into the synaptic cleft. Therefore, there will be not enough excitation of the muscle, isn't it? However, on the myasthenia gravis, the problem is at the postsynaptic membrane, where there is a distraction of the cholinergic receptors. And so when you don't have enough cholinergic receptors, again, this results in what? The same thing, okay? Would be no excitation of the muscle cell. So muscles would be weak. Now, we said that the postsynaptic membrane would be, the postsynaptic potential can be either excitatory or inhibitory, okay? And when we say excitatory, meaning that the excitation would happen by decreasing the voltage, okay, to a less negative value. So, when it goes more into the positive values, isn't it? And inhibition, when you take the post-membrane potential away, or the membrane potential away from the threshold into a more negative values, okay? And so, whether the postsynaptic potential is excitatory or inhibitory depends on many things. It depends on the type of neurotransmitter. If it's glutamate, usually it's excitatory. If it's GABA, it's inhibitory, because glutamate binding into the MDA receptor would allow sodium to go inside the neuron, okay? And this usually brings the membrane close to the threshold and then generating an action potential. Whereas GABA binding into a GABAergic receptor would allow chloride to go inside, and chloride would hyperpolarize the membrane. So it will be inhibitory. Okay, is this clear? Okay, so this is just what I explained. Okay, so like I said, so it will be an excitatory postsynaptic potential when the, for example, with glutamate binding into the MDA receptor, this will create an excitatory postsynaptic potential, which will bring the membrane to the threshold in order to generate an action potential. And so the reason why glutamate would result in an excitatory postsynaptic potential, because it's allowing sodium to go to the inside. And sodium would be going to the inside because the reversal potential for sodium is in the positive values, isn't it? You remember the reversal potential for sodium. Yeah, so it's in the positive values. Whereas the reversal potential, for example, for chloride, it's less than the threshold. So for example, here, you can see here the reversal, if it's this is the reversal potential, for example, for chloride. And as you can see that the outward movement of the, the inward movement of the chloride would make the membrane potential, take the membrane potential away from the threshold. And so this would result in an inhibitory postsynaptic potential. Okay? Is this clear? Good. Okay, any questions? Questions? Okay. Now, another condition, so this reflects how excitatory and inhibitory neurons are important in general, okay? So this is an application for that. So this is a disease, you know, botulism and tetanus. Are you familiar with botulism and tetanus? You know that these conditions are basically caused by toxins from bacteria, which is called Clostridium bacteria. Okay, so these toxins work as basically cleaving. These toxins basically inhibit the neurotransmitter release by cleaving the snares proteins. Remember the snares, which we mentioned in the fusion? Okay, so these toxins would cleave the snares protein, thus disrupt the fusion of the synaptic vesicle with the membrane of the postsynaptic neuron. Now, in botulism, basically, we have a flaccid paralysis, right? Whereas in tetanus, what do we have? Spasm. Why there is a difference here? Although the mechanism is the same. The reason why it's a flaccid in botulism is because the toxins are being targeted to the excitatory motor neurons. Whereas in the tetanus, the toxins are targeted to the inhibitory interneurons. And so when you don't have an inhibition, you have an overexcitation. Okay, is this clear? Very good. Questions? We'll move to the last part. Okay. Okay, so the postsynaptic potential, as we learned, which type of action potential is it? Which type of potential? Is it a graded or is it an action potential?
Graded.
It's a graded, very good. It's a graded. It's not through voltage-gated sodium channels, is it? So once the acetylcholine bound to the cholinergic receptor, it will produce a graded potential. If it's big enough, it will excite the voltage-gated sodium channels, right? Good. Now, graded potential, we know that one of the characteristics of graded potential is that they summit, they can add together, okay? And so a discharge from one single presynaptic terminal usually is not enough to bring the membrane to threshold in order to generate an action potential, okay? So what happens is that the graded potential or the postsynaptic potential will be adding up in order to produce a bigger postsynaptic potential. So you can see here, okay? So let's take this as an example. So we have a presynaptic, an excitatory presynaptic neuron. We have an inhibitory presynaptic neuron, and we have another excitatory postsynaptic terminal, okay? So now, one discharge from either the excitatory 1 or excitatory 2 would generate a postsynaptic potential. But is it enough to reach the, to take the membrane to threshold? It's not enough. So we wouldn't have an action potential. Whereas if we add the postsynaptic potential from E1 and E2, we would bring the membrane to threshold, okay, to generate an action potential, okay? An inhibitory postsynaptic potential, as we learned, it will be taking the membrane away from the threshold. okay? And also by adding the excitatory one with the inhibitory, you're also minimizing the size of the postsynaptic potential. So it won't be able to reach the threshold, okay? Now, there are multiple types of summation. There is the temporal summation and there is the spatial summation. Temporal summation is basically when two synaptic discharges are being discharged closed in time, okay? So they will be adding together. So this is an example of a temporal summation. So close in time, okay, firing from one presynaptic terminal, as you can see here. So we have one presynaptic terminal firing, firing twice in close in time. So they will be adding together to generate a bigger postsynaptic potential. Whereas the spatial summation is coming from two presynaptic neurons, which are happening, which are being firing at the same time, but different location on the postsynaptic neuron. So they will be adding up to generate a bigger postsynaptic potential in order to take the membrane to threshold in order to generate an action potential. Okay, is this clear? Any questions? Any questions? What's the reversal potential for the end plate muscle cell? 0 millivolts. Okay. Cholinergic receptors. So at resting state, when they are bound, when the muscle cell is at resting state, when the acetylcholine is bound to the cholinergic receptor, which ion will flow? Sodium. And sodium would do what? Depolarize. Very good. Depolarizing. Then after the, not depolarizing, it's going to generate a postsynaptic potential. Now, what will happen next? Voltage-gated sodium channels. And this will contribute into the generation of action potential and contraction of the muscle cell or exciting the next neuron. Okay? Good. Thank you very much. Have a nice day. So the irreversible, always 0 for ligand? No, the 0 value is for the neuromuscular junction, for the nicotinic receptors. For this case only. I don't know the reversal potential for other channels. Okay, the spatial summation is basically two discharges, okay, at the same time from different presynaptic terminals happening at different locations within the postsynaptic neuron. They will add up in order to generate a bigger postsynaptic potential. Temporal is from one presynapse, but firing two signals, okay, two discharges. They are coming close in time. They will add. Okay. The reversal potential is basically so the reversal potential is the potential where the movement of ion would be changing from inward to outward. Okay, so for example, in this case, we know that the cholinergic receptors they are permeable to both sodium and potassium. Okay, so 0 is the reversal potential. So when the muscle cell is at rest, so when the muscle cell is at rest, okay, it means that the movement would be inward by entry of sodium, okay, because there is a huge driving force for sodium to go to the inside, because we know that the equilibrium potential for sodium is positive 70. So when the muscle cell is at negative 90 millivolts, sodium will enter inside in order to bring the cell more to the equilibrium potential for sodium. But we know that the cholinergic receptors, they are also permeable to potassium. So at 0 millivolt, it will reverse to an outward movement, which is by potassium. So potassium wants to go out because it wants to reach its equilibrium potential. Okay. I just want to delete
When are you going to upload the lecture?
Sorry?
When are you going to upload the lectures for today and this?
I'll upload them. I thought